Hi, good evening. My name is Gabriela Sanchez. Today is Monday, December 7th, 2020. I'm going to be doing the experiment for week 4. And it's the biochemical testing for microbial identification with the methyl red, Voges, Prosker, and catalase testing. What I'm going to do is I'm going to prepare for the incubation of microbes in the MRVP broth. I have my active broths for E. coli and S. epidermidis, and I also have the MRVP test tubes or the broths for E. coli. One's they're already labeled. One's for E. coli and the other one's for S. epidermidis. I have a small cup of bleach and it is labeled. And I also have 2 pipettes sitting in alcohol. I'm going to light my candle. And this is to disinfect everything before I get started with the cultures. So the first thing I'm going to do is I'm going to start with the S. epidermidis. Actually, we'll start with the E. coli one first. I'm going to start with the E. coli active culture. I'm going to pass the lid or the rim of the test tube by the flame of the candle. This is to sterilize it. I'm going to set this aside. Now I'm going to do the same with the MRVP broth labeled E. coli. I'm going to uncap that. And I'm going to pass it through the flame as well to sterilize it. Next, I'm going to grab one of my pipettes. I'm going to shake it off to make sure that it is dry. I do want to note that I have wiped everything down before starting this procedure. What I'm going to do is I'm going to insert my pipette into my active E. coli culture. I'm going to absorb a little bit. And I'm going to place about 4 drops into my MRVP test tube. So 1, 2, 3, 4. Once I've done that, I'm going to I'm going to put my pipette into the cup that has bleach. Before I recap the E. coli test tube, I'm going to pass it through the flame once more to sterilize it before I recap it. Same thing with the MRVP broth. And I'm going to pass it through the flame and recap. I'm going to do the same thing with the S. epidermidis active culture. I'm going to uncap, pass it through the flame to sterilize it. I'm going to do the same with the MRVP broth. Uncap it, pass it through the flame. Now I'm going to grab my other pipette, shake off all the excess alcohol, grab a little bit with my pipette from the active culture and place about 4 drops into the other into the MRVP. And I've just placed that in the bleach. Before I recap it, I'm going to pass it through the flame once more to sterilize it before I recap it. Same with the MRVP broth that's labeled S. epidermidis. Pass it through the flame and recap. So I have done the same with both active cultures, the E. coli and the S. epidermidis. Once I'm done with this, I'm going to wipe everything down, discard whatever needs to be discarded, and I'm going to let this incubate for about 48 hours. I'll make a results video at that time. Thanks.